Where does synthesis of fatty acids occur? In the cytocell. Fatty acid synthesis utilizes blank, whereas degradation utilizes coenzyme A. Acyl Carrier Protein. True or false? NADH is used for both synthesis and degradation of fatty acids. False. NADPH is used for synthesis. Enzymatic activities of synthesis are combined in one large polypeptide called blank. Fatty acid synthesis. Carbon dioxide, in form of bicarbonate, is added to acetyl-CoA in the initial step of lipid synthesis to form what? Malonyl CoA. What are the two principal biologically active forms of pantothenic acid? Coenzyme A greater than fatty acid degradation, acyl carrier protein greater than fatty acid synthesis. What are the reactants and products of round 1 of fatty acid synthesis? Reactants: acetyl ACP and malonyl ACP. Products: butyryl ACP. What happens in round 2 of fatty acid synthesis? Same enzymatic activity, only butyryl ACP now condenses with malonyl ACP. What is the final step of fatty acid synthesis? Cleavage by thioesterase. True or false? Thioesterase acts as a ruler to determine fatty acid chain length in biosynthesis. True. Fatty acid synthesis occurs in cytosol, but acetyl-CoA made in blank. The mitochondrial matrix. What intermediate carries acetyl groups from mitochondria to cytosol for fatty acid synthesis? Citrate. Synthesis of malonyl CoA is a reaction catalyzed by blank, a key regulatory in fatty acid metabolism. Acetyl-CoA carboxylase. Acetyl-CoA carboxylase is blank by phosphorylation and blank by dephosphorylation. Switched off, activated. True or false? Glucagon and epinephrine stimulate the activity of acetyl-CoA carboxylase, while insulin acts to inhibit the carboxylase. False. The opposite is true. Essential fatty acids must come from blank. Diet. Mammals lack enzymes to introduce double bonds beyond which carbon on a fatty acid chain. C9. These stimulate smooth muscle, regulate blood flow, adhesion of blood platelets, inflammation. Prostaglandins. Aspirin, an anti-inflammatory, blocks blank synthesis. Prostaglandin. Where are prostaglandins synthesized? On smooth endoplasmic reticulum. Which intermediate is common to both triglyceride and phospholipid synthesis? Phosphatidate. True or false? In phospholipid synthesis you can have synthesis from an activated diacylglycerol, phosphatidyl serine, inositol, synthesis from an activated alcohol, phosphatidyl ethanolamine, choline. True. What is the most abundant phospholipid in mammals? Phosphatidylcholine. Phosphatidylcholine is also a major blank blank. Pulmonary surfactant. What are plasmalogens? Where is choline plasmalogen found? Glycerol ether phospholipids, abundant in cardiac tissue. Phospholipid initially isolated from beef heart, present exclusively in her mitochondrial membrane. Cardiolipid or diphosphatidylglycerol. Sphingolipids are synthesized from what? Ceramide. In sphingolipid synthesis, two fatty acid chains added to small amino acid blank rather than two 3-carbon glycerol backbone. Serine. Blank are carbohydrate-rich sphingolipids that contain acidic sugars. Gangliosides. The very property that makes me useful in cell membranes, namely my absolute insolubility in water, also makes me lethal. What am I? Cholesterol. True or false? Sphingolipid modulates fluidity of cell membranes and a precursor of steroid hormones. False. These are characteristic of cholesterol. Enzyme in synthesis of cholesterol, blank, target for cholesterol-lowering drugs, histidines. HMG-CoA reductase. What is bad cholesterol? Low-density lipoproteins. True or false? High-density lipoproteins pick up cholesterol from dying cells and transport back to the liver. True. Which of the following substances represents the immediate precursor for fatty acid synthesis? A. Glucose, B. Citrate, C. Acetyl-CoA, D. Acidoacetate, E. Beta-hydroxy-beta-methylglutenyl-CoA. C. Acetyl-CoA. Pantothenic acid is an integral part of A. NAD, B. Cobalamin, C. Folic acid, D. Coenzyme A, E. Pyridoxine phosphate. D. Coenzyme A. Which of the following statements is true concerning fatty acid synthesis and fatty acid breakdown? A. Both are mitochondrial functions. B. Both require phosphopantothenic acid. C. Both share common electron donors and acceptors. D. Both share common enzymes for oxidation and reduction reactions. B. Both require phosphopantothenic acid. The pathway of extramitochondrial synthesis of even-numbered fatty acids differs from that of the catabolism of fatty acids in that A. malonyl-CoA is an intermediate in synthesis, B. acyl carrier protein is needed in catabolism. C. No flavoprotein enzymes are required for catabolism. D. Propionyl CoA may serve as an intermediate in synthesis. A. Malonyl CoA is an intermediate in synthesis. Carbon dioxide or bicarbonate is required in the biosynthesis of fatty acids because C. Carbon dioxide is incorporated into acetyl coenzyme A, forming malonyl coenzyme A, an intermediate. In the synthetic process, the first step in the pathway for fatty acid synthesis is catalyzed by an allosteric enzyme, which is the principal regulator of the pathway. This enzyme is A-thiolase, B-pyruvate carboxylase, C. citrate synthetase, D-acetyl-CoA carboxylase, E-pyruvate dehydrogenase. D. acetyl-CoA carboxylase. Prostaglandins are made within cells, A on an RNA template, B on rough endoplasmic reticulum, C from methionine, D from progesterone, E from polyunsaturated fatty acids. E from polyunsaturated fatty acids. Complete hydrolysis of 1 mole of lecithin yields Al amino acids, Bd monosaccharides, C phosphatidylethanolamines, D2 fatty acids, 1 glycerol, 1 phosphoric acid and 1 choline. Residue, E2 fatty acids, 1 glycerol, 1 phosphoric acid and 1 cholesterol molecule. D. 2 fatty acids, 1 glycerol, 1 phosphoric acid and 1 choline residue. Gangliosides are glycolipids found on various cell surfaces. In addition to sphingosine, their unit composition contains another characteristic component. This component is A. uronic acid, B. plasmalogen, C. triglyceride, D. N-acetylmuramic acid, E. N-acetylneuramic acid. E. N-acetylneuramic acid.